Hi everyone, my name is Isabelle, and together with Beatriz and Natália, we'll be bringing a few considerations about the gastrointestinal implications in the new coronavirus. Coronavirus disease 2019 was first reported in Wuhan, China, during December 2019. It is a newly emerged viral infection caused by the severe acute respiratory syndrome coronavirus 2. It initially produced a pneumonia outbreak in China and then quickly spread across the globe. In January, the World Health Organization declared the epidemic to be a public health emergency of international concern. COVID-19 has mainly gained attention for its rapid contagious ability and severe respiratory manifestations. The coronavirus pandemic is considered a global health crisis of our time and the greatest challenge we have faced since World War II. Today, we count more than 24 million confirmed cases and almost 822,000 deaths reported worldwide. The disease is characterized by fever, dry cough, fatigue, and lymphopenia, which can also lead to gastrointestinal symptoms, organ dysfunction, rapid progression to a severe acute respiratory syndrome, and even death. Based on recent publications, 1, 2, 3, the most common symptoms in patients infected by the 2019 nCoV were fever and cough. However, the incidence of other clinical features differs in different reports. Here, we would like to bring attention to the gastrointestinal manifestations. Two other unusual coronavirus outbreaks occurred in the 21st century, including the severe acute respiratory syndrome coronavirus here and the Middle East respiratory syndrome coronavirus, causing high mortality rates. As the seventh member of the coronavirus family that infects humans, the 2019 nCoV genome sequence is reported to be approximately 82% identical to the human SARS-CoV. Besides leading to similar symptoms, the 2019 nCoV has the same cell entry receptor as the SARS-CoV, the angiotensin converting enzyme 2, ACE2, which regulates the cross-species and human-to-human transmissions of the SARS-CoV. So, let's talk about it. The enzyme ACE2 is considered a major functional receptor of the SARS-CoV-2 in humans. It is distributed broadly, which can indicate that COVID-19 may involve multiple organs and may explain the various extrapulmonary symptoms. Studies show that the protein expression level of ACE2 is significantly higher in the small intestine than in any other tissue. These results suggest that cells of the small intestine may be potential target organ of the virus. But how does it happen? The virus spike protein enters the small intestinal enterocytes by binding to ACE2, which induces immune cells infiltration and abnormal intestinal function. The dysregulation of ACE2 modulates innate immune responses and contributes to gut dysbiosis. This also may explain diarrhea observed. A study showed that among the symptoms, diarrhea displayed the smallest criteria value, suggesting that the criteria for diagnosing diarrhea may differ in different hospitals. Due to the different criteria, clinicians may underestimate the value of this symptom in clinical practice, and it may affect the preliminary diagnostic accuracy. Previous studies of the 2003 SARS outbreak in China showed that diarrhea is a common presenting symptom of SARS. The intestinal tropism of the SARS-CoV has major implications on clinical presentation and viral transmission. Also, a high virus titer in the digestive tract may indicate a high risk of transmission via feces of the patients, which also raises new problems in the disposal of feces.
It is reported that diarrhea may be underestimated in COVID-19 patients. A study found more than a half of the hospitalized patients tested SARS-CoV-2 RNA positive in stool samples, while no more than 20% of confirmed patients were reported with digestive symptoms in other cohorts. Also, the research found positive staining of ACE2 and SARS-CoV-2 in gastrointestinal epithelium from patients with positive SARS-CoV-2 RNA in their stool, which added convincing evidence that the COVID-19 can infect the digestive system. It is speculated that persistence of gut infection caused by SARS-CoV-2 can lead to readmission at the first discharge or time of waiting for discharge. In this view, another paper reported 3 SARS-CoV-2 confirmed patients with persistence of gastrointestinal symptoms and SARS-CoV-2 RNA in stool samples on readmission after SARS-CoV-2 pneumonia had resolved. How SARS-CoV-2 affects the human lungs and gut? SARS-CoV-2 can be transmitted through the respiratory or digestive tract, directly infecting the host by binding with the ACE2 receptor of pulmonary epithelial or intestinal epithelial cells. This leads to lung and/or intestinal tissue damage and a systemic immune response. However, after initially infecting the lungs, SARS-CoV-2 can break through the mucosal immune barrier and indirectly affect the gut along the gut-lung axis and vice versa. Gut tissue damage, an excessive inflammatory response, and a dysfunctional immune response can lead to gut microecological disorder. However, some interventions, such as probiotics, beneficial metabolites, and eliminating harmful bacteria, can provide resistance against these adverse effects. In case of a pathogenic SARS-CoV-2 infection, a healthy intestinal microbiota could be essential in maintaining an optimal immune system to prevent excessive inflammatory responses that eventually become detrimental to lungs and vital organ systems. The gut microbiota might regulate host immune and inflammatory responses along the gut-lung axis through microbial metabolites and the mucosal immune system. The figure shows a possible model for using probiotics and beneficial microbial metabolites against lung infection and injury. Probiotics and SCFAs can be taken up by M cells and presented to T cells as antigens via dendritic cells, leading to T and B cells proliferation and activation. Guided by immune mediators, immune cells are then localized at the lung infection site, enhancing antiviral immunity and providing protection to the lungs. The gut innate lymphoid cells can migrate to the lungs to enhance antiviral immunity via lymphatic and blood circulations. Surface IgA can be produced and transported from gut-associated lymphoid tissues, GALT, to the surface of the pulmonary mucosa, which can prevent virus adhesion and consolidate the mucosal barrier. SCFAs produced by intestinal microbiota can be transported to the lungs through the blood, where they can play an anti-inflammatory role and consolidate the lung mucosal barrier. SCFAs can also be transported to the bone marrow and enhance its hematopoietic function, further promoting the proliferation and activation of dendritic cells and other immune cells. Overall, this phenomena can enhance the antiviral immunity of the host.
As the largest immune organ of the human body, the gut harbors approximately 10 to the power of 14 bacteria. The gut microbiome is an indispensable functional organ and participates in immune response regulation, nutrient absorption and metabolism, and the control of infection. However, the homeostasis between the gut microbiome and the host immune system is impaired as viral infections, antibiotic use, and chemotherapeutic applications increase. ACE2 downregulation in SARS patients during infection decreases the gut absorption of tryptophan and reduces the secretion of antimicrobial peptides, leading to increased pathogen survival and gut dysbiosis. Therefore, the ACE2 dependent regulation of the microbiota may explain the occurrence of diarrhea in SARS-CoV and SARS-CoV-2 infections. Microbiota dysbiosis is the abnormal shift in the number, species, and proportion of normal microbial populations in the gut, which results in an abnormal physiological and pathological combination. In a particular study, significant change in the gut microbiota in a cohort of patients with COVID-19 was observed. Lactobacillus and bifidobacterium produce lactic acid and play an important role in regulating immunity and maintaining gut barrier function, and its number would decrease in the patients with COVID-19. Butyrate plays an important role in inhibiting the overgrowth of opportunistic pathogens, maintaining the integrity of the intestinal mucosal barrier, activating the adaptive immune response, and enhancing antiviral immunity, and butyrate-producing bacteria was also decreased in these patients. Plus, an overgrowth of opportunistic pathogens such as Enterococcus and Enterobacteriaceae was observed, which are involved in the disease severity. Preventing bacterial translocation from the gastrointestinal tract is a priority in the treatment of COVID-19 and associated complications. Prophylactic antibiotic therapy at the expense of maintaining a balance in the gut microbiota is a risk factor for secondary bacterial and fungal infections. Microbiota-targeted therapies have not currently received enough attention in the study of acute infectious diseases. Microbiota-targeted therapies administered at the early stage of COVID-19 may help decrease the number of severe and critical cases. Probiotics are defined as being living microorganisms which, when consumed in sufficient quantities, provide health benefits to the host. Recent studies have demonstrated the effects of probiotic bacteria on the immunomodulatory capacity through the modulation of the gut microbiota, which can help fight infection by COVID-19 and reduce the risk of secondary bacterial infection due to prolonged antibiotic exposure in several experimental COVID-19 treatments. The COVID-19 pandemic presents a significant social and economic burden worldwide, and studies on the disease unfortunately remain at a very preliminary stage. The structure and function of the gut microbiota could be a potential biological mechanism behind the diverse susceptibility of different groups of people to SARS-CoV-2. However, the specific mechanisms through which the SARS-CoV-2 infection influences the intestinal microbial community among patients of different ages, ethnic groups, and geographical locations require further investigation. It is worth noting that the rationale for using probiotics in COVID-19 is derived from indirect evidence. Different kinds of probiotics and/or different doses of probiotics usually have different biological effects on the host. To further understand the specific mechanism and elucidate the benefits of personalized function food including probiotics and metabolite products in the prevention and treatment of COVID-19, more clinical trials and evidence-based medical data are needed. We appreciate your attention. Stay home if you can and stay safe. Bye!